yesterday we were speaking about, we have started to discuss about bio or about affinity biosensors, and yeah, and one of the most used affinity biosensor is the antibody and antigen biosensor, or short we call as immunosensor. Biosensor that use immunoagent as their biosensing element. So, and this make use of the high association constant between the antibody and antigen, and the antibody and antigen complex. Uh, the affinity is measured as polar association constant. Change of the antigen for a corresponding antibody, which is measurement of the binding forces in the resultant ADAB complex, is expressed as a polar association constant. Yeah, so this is expressed like this reaction sim. Yeah, so it is case between the antigen antibody, there's a KF, the formation with association constant to make AGAB complex, and basically it can goes back to the dissociating to into the original antigen and antibody molecules. The ratio between KF and KR here is the Ka, the association constant. Yeah, Kf is the forward rate of the complex formation. Kr is the reverse rate for the dissociation constant. So when the Kf is much larger than Kr, then we have a very high Ka, or means then we have very strong binding between the antibody and antigen specific. The vast majority of antigen-antibody interaction under conventional pH, and conventional pH means uh, 6 to 7 in the neutral region under conventional temperature at room temperature 25 degrees Celsius and buffer conditions. Kf much larger than Kr will give a very high value of Ka. Yeah, and the Ka here is up to 10 to the power of 15 mole per liter. So this is a very big value for Ka. The Ka has been measured. So very strong. And this is in contrast with enzyme, for example, which is quite reversible. Here, majority of enzymatic Kf value or protein affinities are in the range of 10 to the power 3, 10 to the power 6 mole per liter. So we can see here the binding between antibody and antigen is about 1 billion, 1 billion times larger than the affinity between enzyme and its substrate. The much greater magnitude of Kf relative to Kr for antigen antibody interaction is a consequence of the highly optimized multi-site attractive forces between antigen and the binding pocket of the antibody. Okay. So that is why it is not easy. It is very good, very selective, very sensitive, very specific if we use immunosensor, but they are usually not reversible. So after they bind to the corresponding analyte, usually you have to dispose them. Very difficult to make them separate. Okay? And this is one advantage, but also disadvantage, because until now maybe we want for portable and one-time use biosensor, it is good enough. But there is an increasingly new field of application like wearable device, wearable sensor. Yeah, if the wearable biosensor is made from antibody, for example, then it means every time you have measure one time, you have to dispose them. It is not practical. Okay, so that is why uh, 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 different kind of biosensors need to be used. Now, the idea is how can we know how many of this binding occurs? Yeah, this binding occurs to in order that we can detect the binding, we need what we call as tracer. Yeah, tracer for to generate the signal. As to measure then the concentration of the analyte, the antigen requires the presence of signal generating tracer in the immunoassay format. A tracer is used to generate signal. It could be optical, yeah, could also be electrochemical. So optical means that, for example, you label the antigen or you label the antibody. Yeah. So, for example, when it binds to antigen, but if you label all the antibody that is being immobilized onto the surface of the substrate, when they bind to antigen, the fluorescence, for example, decrease. Yeah. So then you know how much of them are being bound to the antigen. Otherwise, you do what we call a sandwich immunoassay. What is sandwich immunoassay? So this is the substrate. We mobilize the first antibody, like this, and then afterwards come the antigen, bind to it in a specific binding site, monoclonal. Then we put secondary antibody, yeah, that bind to the antigen at another specific site. And also with this secondary antibody, we put a label. For example, it is attached to enzyme that can fluoresce or luminescence. Yeah. So this is commonly called as enzyme ELISA. Yeah, ELISA. ELISA. This ELISA is enzyme link. Here, immunosorbent assay. So because the second antibody is linked with an enzyme that can label the reaction. So then you can put the whole reaction chamber into a fluorescent reader, for example, or luminescent reader. Then you can quantify the amount of antibody that bound to that antigen, yeah, with where also that it also shows the quantity of the antigen. Okay. So it could also, yeah, this one, fluorescent molecule, fluoroimmunoassay, or in the form of enzyme immunosorbent assay, or could also be radioisotopes, yeah, where it is then you use a Geigen counter, for example, to detect the amount of radioactivity. Okay, okay, so then drawing. So the immunosensor is essentially to develop more simple antibody antigen tests, yeah, compared to immunoassay, which combine the great specificity and sensitivity of laboratory-based assay, but with simplicity of a biosensor. So it depends, yeah. If it were like color change, then we can quantify easily now using camera or even using bare eye. If it is electrochemical immune sensor, then we can detect we can detect using electrodes, for example. If it were if the use is we can label the antibody or antigen using a mediator, similar like the previous last yesterday session with a mediator, then we can detect the electron exchange with the electrode. Alternatively, we can use a technique called as impedance, electrochemical impedance spectroscopy, where we measure the impedance, the resulting impedance between the between the two electrodes, yeah, caused by that binding reaction. So when it is only antibody, of course, then the impedance will be different when it already bound to an antigen. Okay. So yeah, like this, usually we have, this is what I have just drawn in the previous page. Yeah, we have a secondary antibody. So first antibody is bound to a substrate, what we call as a primary antibody. The sample is then introduced to the device, bind to this antibody, to the primary antibody, and then we introduce a secondary antibody, or yeah, antibody is one, yeah, secondary antibody, which is labeled with whatever it is. It could be fluorescent labeling or enzyme labeling or radioactive labeling. So upon binding, yeah, so you see here that the two antibodies have different activity towards different binding sites, yeah, of the antigen. So upon binding, then the signal will increase. Yeah, the more antigen is bound to the primary antibody, of course, then the more signal you will have. Then this is what we call as competing. So in order to reduce the complexity of having to introduce a secondary antibody, what we can do is also to introduce similarly antigen which is labeled already, which is labeled. So this will introduce at the same time with the antigen from the sample. Okay, so then they will have a competing reaction with the primary antibody bound to the substrate. When the concentration of the unlabeled antigen is more, is higher, the higher it is, then the more of the unlabeled antigen will be binding, will be bound to the antibody, resulting in a decrease of the signal, whether it is electrical signal or fluorescent signal or radioactive signal. Okay? But when there is no antigen at all, the respective antigen at all at the sample, then what we will have is only the labeled antigen, which is already prepared from the lab, from the with the that biosensor also. Okay, so then the sensitivity, the signal will be maximum. So the higher the concentration of the antigen at the sample solution, then the signal will decrease more. Okay, so this is what we call as competitive assay. The first is labeled sandwich immunoassay, because it leads to antibodies which is sandwiching the antigen of interest, and the second is competitive assay. Okay. The signal is proportional to analyte concentration. This in the former, in this sandwich immunoassay, and while this one it is inversely proportional, in the competitive immunoassay. Okay, now what if we don't use any label? How can we detect also the binding between the antibody and antigen? So here still we need to prepare a labeled antigen. But what if we think it is tedious? Let's just have the antibody being bonded, immobilized onto the substrate, and then introduce the antigen. There are many techniques that can be used, to detect such binding. I have mentioned electrochemical impedance spectroscopy, right? But we have many other techniques that can measure mass changes on the substrate. One of them is called as SPR-based immunosensor. So SPR is surface plasmon resonance. Surface plasmon resonance based biosensor system. So this is based on the electron resonance at the surface of the biosense of the transducer itself. Usually it is made from metal thin film of metal, preferably gold or silver, and the electron you see from from biomaterial, the electron in metal, they have electron cloud, and so they are freely flowing from one atom to another, right? When you introduce light at certain angle, it will resonate at certain frequency, and then part of this incoming light, you have to be at certain angle. The incoming light will be absorbed by these free electrons, to resonate in themselves. So if then you capture the reflected light, they will have absorbed intensity, okay, at certain angle. Yeah. So that is what we call as surface plasmon, resonant plasmon. Okay. So this is related with the refractive index and the dielectric constant of the surface. Okay, and then now it's already commercially available, albeit they are still very expensive. So it will be interesting if we can make it ourselves. So this is presents significant breakthrough in molecular sensor, particularly used in immunosensor techniques. So it have shown promising in providing direct measurement, yeah, without having to label the sample. Yeah. So when this one, yeah, SPR is a mechanism, a phenomenon which occurs when a beam of light is directed onto a glass metal interface, usually a glass prism, on a gold, silver, or metal layer at a specific angle, yeah, only at a specific angle, which is the resonant angle. A component of the electromagnetic light wave will propagate in the metal along the plane of the interface in the form of surface plasmon. Okay, then the resonant angle is sensitive to the changes in the refractive index and dielectric constants at the interface up to 1000 nanometer from the actual metal surface. Yeah. So therefore, when anything happens in the surface, for example, due to the binding reaction between the antigen and the body, of course, the refractive index will change. The dielectric constant also will change in the surface. And this will cause a change of the resonance pattern. Yeah. So immobilization of the antibody on the surface causes a measurable shift in the resonant angle. On binding of silver to immobilized antibody, a further change will occur. Yeah, this binding induces shift in resonance angle, expressed as resonance unit, linearly proportional to the concentration of bound antigen or antibody if the antigen is pre-immobilized, depending on which system you are making. This is how it looks like in schematics, yeah, of the system. So you have a light source here, okay, put on through a prism. I believe you have learned in physics that it will be then diffracted by this prism, yeah, into various spectrum of wavelength. Okay, it is reflected by the sensor surface here with gold film, either gold or silver. Usually gold or silver being used a lot, yeah, as for SPR. Then on top of the gold thin film, you immobilize the biomolecule. If it could be like this, antibody, and when we flow the antigen, some will bind, and this binding will change the refractive index and the dielectric property of the surface, which will make a shift, causing a shift in the resonance pattern. So this is the sensorgram. So at the beginning, it was absorbed at this angle, yeah, but then upon binding of of the antigen towards the antibody, it will shift the curve to this second curve. Then the shift, yeah, can be depending on the concentration of the antigen. The shift will also be different, and then we can plot the shift on the left side, on the y-axis, and concentration on the x-axis. Then we can have a calibration curve of shift resonance unit, yeah, shift of resonance unit as a function of the concentration. This is how it works. Okay, so this one is another one from SPR immunosensor using polarized light source and using photodetector like this. And when there is association here, then dissociation, it can measure this, yeah, and the binding like this. This is one, and there is this regeneration, and there is dissociation here. It drops back again. So this is a SPR immunosensor from Dutra and Kubota. Yeah. To determine CTNT, cardio-specific marker, highly sensitive marker for myocardial damage, released into bloodstream during acute myocardial infarction with a detection limit of 0.01. Remember what the definition of detection limit? Yeah, limit of detection is basically a resolution similar to resolution. In 5 minutes, time response. Okay. Okay. Another way, that is optical way. Yeah, during optical way. Another way is using mechanical way, which is using mechanical vibration. Remember in the discussion about piezoelectric sensor, we discussed about QCM, quartz crystal microbalance made from piezoelectric material. Piezoelectric material is when you apply a varying voltage, yeah, then it could vibrate with certain resonance frequency. So the piezoelectric sensor made from piezoelectric effect, for example, piezoelectric quartz, yeah, quartz crystal microbalance, QCM. And this is quite widely used and implemented around the world. Yeah, this QCM sensor. The sensor is based on the principal theories of close relationship between. This is the important mechanism. The resonance frequency is dependent on the mass loading. Okay. So when the mass loading change, the resonance frequency will also change. That's why when we can immobilize antibody on the surface of QCM, so upon binding to the antigen, then resonance frequency will also shift. Yeah. Okay, so that's why the such piezoelectric sensor is also called as quartz crystal microbalance QCM. And this has been widely used. Yeah, for example, here using ET-cut piezoelectric quartz crystal film with cold electrodes are deposited on both surfaces. Okay, and like this, the delta F, this is the shift, yeah, is given in relation with the mass change, or the expression between the frequency shift and the mass change given by the Southbury equation. By the Southbury equation, like this, delta F equals to minus 2F squared divided by square root of rho Q mu Q multiplied with delta M. This can be shortened as a constant, yeah, minus CF times the delta M. So the more antigen, for example, binds to the antibody, more complex are being formed, then the mass change also will increase. So then also the frequency will shift even more. Okay, so delta F is frequency shift resulting from the additional mass per area. F is the intrinsic crystal frequency. Rho Q is the density of the quartz, which is the shear modulus of the quartz film. So this is how the system look like. This is a quartz wafer. You put the gold electrode, yeah, like this, to apply the varying voltage as well as to measure the frequency shift. And you oscillate it using oscillator circuit onto the piezoelectric crystal to flow the sample onto it, yeah. And you count the frequency from this oscillator circuit. And if the frequency shift, it means then something is adsorbed on the surface of the piezoelectric crystal. So, for example, here, this is the original unloaded QCM. When you introduce a sample that is adsorbed on the surface of that QCM, there is a delta M. And this, due to this delta M, according to the Southbury equation, it will create a shift delta F of frequency, and the resonance will shift. Yeah, if then we plot the frequency as a function of time, yeah, the resonance frequency is a function of time. Yeah, upon analyte addition, upon the addition of analyte, see the frequency will shift, will go down. But if you introduce other thing, for example, that can be binding to the first analyte, it will create another dip of the frequency. Yeah, so this is how QCM works. Okay, then therefore we can apply it. For example, here this is using this is gold surface and using gold as we are binding antibody onto this linker, kind of linker, cross-linker molecule. We are binding the biomolecule, the antibody onto the gold surface, yeah, so that can detect salmonella, yeah. Oh, sorry, E. coli to the surface of E. coli. One type of QCM immunosensor. Okay, those are immunosensor. Yeah, we presented two examples of the physical transducer, the surface plasmon resonance and the quartz crystal microbalance. There's also another one, electrochemically, using EIS, electrochemical impedance spectroscopy, which can also be used to detect binding. Okay, then nucleic acid biosensors. Okay, the nucleic acid biosensor, like this. This depends on the recognition, yeah, due to the hybridization of the deoxyribonucleic acid or ribonucleic acid. Yeah. So this is the biorecord. And we know that the recognition is very specific. Thymine only binds to adenine, yeah, and cytosine only binds to guanosine. Okay. Yeah. So the complementary binding for this is the form the basis, yeah, of the specificity of biorecognition in DNA biosensor, also referred to as the genosensor. Okay, also referred to as the genosensor. Like this is how DNA look like, a double helical structure and with a specific complementary binding. So when we want to detect a specific sequence of DNA, for example, like let's say for SARS-CoV-2 virus, then we have to know which gene specific to that organism, in this case SARS-CoV-2 virus. Then we apply the single strand specific recognition layer onto a substrate that will bind to the complementary single strand sequence of that gene. Yeah. So when there is binding, and there will be signal transduction, then that should be electronically readable. We can also use SPR, yeah, to detect optically, or we can use QCM to detect using mechanical frequency, mechanical vibration frequency sieve, or using EIS as well. Or we can use label or a mediator to detect directly electronic exchange due to this binding. Okay, like this. This is using chemical light, yeah, like attending. So nucleic acid sensor for fast and easy, fast, reliable DNA testing. This is using QCM, okay, using frequency counter oscillation circuit. The single strand, yeah, single strand DNA probe is bonded to the gold surface onto the QCM using sulfur or thiol, is commonly called as thiol, the sulfur functional group, yeah, bound to this what do you call it, phosphate, yeah, phosphate functional group, and then bind to the 5' of the single strand DNA. When the target DNA exists or occurs from the introduced solution, introduced sample, then it will bind specifically to this single strand DNA, okay, and this will create an additional mass to the surface of the QCM. And then, as we have learned from the previous slide, from previous page, that the frequency will decrease as a function of time. Okay, so then with this sensor, we can specifically detect what strain, for example, of bacteria or what strain of virus, for example, appear on that sample. Okay, and Okay, because of the time, I will just try to sweep it quickly. Yeah, so we only can cover around one one fourth, a quarter or one third of actually the whole chapter. Yeah. So yeah, these are the advantages of the sensor. I will put this onto the Google Classroom so that you can also have a read, yeah, in your lecture time as an additional knowledge. But bear in mind that the content of this textbook is about 10 years old. So it is written in the first decade of the 21st century. So now a lot has changed. Not yet, the basic principle are still the same, but many new techniques, new principles are being introduced. This is a sensor using receptor and ion channel, yeah? Using bilayer lipid membrane similar to the cell membrane. Then to have the receptor onto it, yeah, so that then we can recognize also particular ion or particular small molecule. Okay, then you have receptor channel and ion channel, and electrifying cell receptor, and this is a more larger scale sensed by sensor using cell and tissue. If we would like to recognize mechanism at cellular level, for example, we want to detect the toxicity of certain drug, a particular cell type, then we can use cell or tissue biosensor. Yeah. So similar to whole microorganism sensor, like we have explained yesterday. Yeah, but now instead of using bacteria, we use a cellular metabolism, usually from a mammalian cell, animal cell, like this. Yeah. So, and we need to detect a certain biomolecule, a certain aspect which require a lot of metabolism that finally will result in detectable molecule. Then usually we need to use this whole cell sensor. Okay, then this is microphysiometer. It's quite very specific. I will not explain it. This is then cellular impedance biosensor. I think when we did lab visits to Taiwan, yeah, remember the Professor Lee, Thomas Lee, he made mention about the development of impedance and then to immobilize neurosensor on top of that electrodes, yeah, to measure their impedance for cancer therapy screening. So this is basically that kind of the theory of behind that kind of sensor, yeah. So here we have electrode array, and then the cells are being immobilized onto it, and then we measure the impedance, yeah, change due to when the sensor proliferate or when the sensor dies, then the impedance will change also accordingly. Okay, this is the model of the cell impedance. Okay, cell impedance sensor like this. The application is for example, drug screening, yeah, like this. When the cisplatin here introduced at different concentration and control, how will the cell react? And this is shown as the change of impedance level. Extracellular, it is similar basically. The measure extracellularly. Yeah. Okay. And the biochips is similar to the discussion about the lab on chip here, macrophysics. I believe we have discussed it quite a lot in the previous chapter. Okay, I think I would conclude the biosensor chapter on that. Yeah. So please read again later the chapter. As a final marks, it will also conclude the